Hello friends, welcome back to another video tutorial from Arun's Biology. In today's lecture, we'll discuss about the Hershey-Chase experiment. So let's begin the topic. Hershey-Chase experiment was performed by two eminent scientists, Alfred Hershey and Martha Chase, in the year 1952. This experiment provides an unequivocal experimental proof that supports that DNA is the genetic material but not the protein. This experimental procedure is basically based on the transduction of bacteriophage in the E. coli bacterial cell. Here two different animal models were used. One is the bacteriophage and another is the E. coli bacteria. The T2 bacteriophage is basically a virus and it infects the E. coli or Escherichia coli bacteria. Now coming to the experimental procedure. First, labeling of the protein code, that is also known as the capsid of bacteriophage, and the DNA, that is the genetic material of bacteriophage, was performed. They have taken two different culture medium and cultured the bacteriophages. One culture medium was supplied with the S35 radiolabeled sulfur and it get incorporated into the protein as being a part of amino acid, because sulfur is present in some amino acids like the cysteine or methionine. So it get incorporated. As a result, the bacteriophage's protein code is got labeled with the sulfur. Whereas in the second culture medium, they have provided the P32, that is radiolabeled phosphorus, and radiolabeled phosphorus being a part of the DNA, it get incorporated into the phage DNA. Now coming to the steps of the experiment. There are basically three steps in Hershey-Chase experiment. First is the infection. Okay, so they have allowed the radiolabeled bacteriophage in two different cultures. So there are two different culture procedures. So they have allowed this bacteriophage to infect the Escherichia coli cells. Okay, and the phage have transferred its genetic material into the E. coli cell. In the second step, which is called as blending, with the help of mechanical agitation or blending, they have separated the bacteriophage that have already transferred its genetic material into the E. coli cell from the E. coli itself. Okay. In the third step, they have applied the centrifugal force so that the heavier E. coli cells get precipitated and form the pellet, while the lighter bacteriophage cells remain in the supernatant. Okay, now coming to the conclusion part. After the centrifugation, they got the pellet in the first case where it was supplied with the S35 and the protein code was radiolabeled. They did not found any radioactivity in the pellet, but radioactivity was found in the supernatant, that means in the bacteriophage capsid that have already donated its genetic material. While in the second culture, they got the pellet with radioactivity, because the genetic material of this bacteriophage has been transferred into the E. coli cell and it got incorporated into the bacterial genome, and during this centrifugation, the bacterial cells are heavier and it formed the pellet, and along with that, the radioactive DNA that was actually present in the bacteriophage was also come in the pellet. So radioactivity was found in the pellet but not in the supernatant. From this, the Hershey and Chase concluded that P32 as a part of phage DNA transduced to the E. coli cell and it get incorporated into the bacterial genome, and it appears in the pellet as radioactivity. Thus, it can be said that DNA is the genetic material but not the protein. Okay, so this experimental proof actually provides an unequivocal experimental support for the DNA as the genetic material. So this experiment is very much important for the board examination purpose. So prepare all the 3 steps of this experiment. And one main important thing that sometimes asked in the examination is why this blending is being performed. Okay, so what is the purpose of this blending? So blending is basically performed so that this bacteriophage gets separated from the bacterial cell. Otherwise, it remain attached with the bacterial cell. As a result, both of them come to the pellet. So if this bacteriophage, which is labeled with the S35, will come with this pellet, and this pellet will also become radioactive in nature. Okay, so in both the culture medium we will get the radioactivity, which is sometimes called as the false positive result. Okay, so to restrict this false positive result, we have to perform the blending experiment. Okay, blending experiment. So this blending is basically done so that this false positive result can be inhibited. Okay, so this is all about the Hershey-Chase experiment. In the next video lecture, we will discuss about the replication of DNA. Thank you for watching.